Hello, everyone. My name is Marco. I'm working at Oliveira's synthetic biology group at Embrapa here in Brazil. And we've been working with integrases in the minimal cell for a couple of years now. And this time I'm not going to repeat the results. I'm going to talk a little bit about the things we're planning to start doing now with them. So we can expand the applications of such serine-integrase in the model. So integrases, they are recombinases that they are able to rearrange DNA sequence upon attaching to specific ATT sites. There are attachment sites named as ATT B and B, and their advantage, especially to use in a minimized molecular environment like the minimal cell, is that they are able to attach the DNA, perform double-strand breaks, rearrange such DNA strands, and then perform the ligation without the need of any other cofactor from the host or repair machinery, for instance. And so we already was working in other models with these molecules, and we decided that would be a good candidate to work on the minimal cell. And the thing that's interesting about their mechanism of working is that depending on the way you design their attachment sites, you can have different outcomes. So the main ones would be integration, when you have ATT B and P on different molecules. And upon their recombination, you have the integration of the circular external DNA into your main molecule, or a genome, for instance. You can have excision when both sites are in the same molecule and facing the same orientation. That's an important detail. When you have the recombination, you're going to have an excision of the circular molecule and formation of the recombined ATTL site on as like working as a scar on your original molecule. And you can have inversion when you have the same molecule, both sides, but now they are facing each other instead of the same orientation. And it has an interesting application, because when you have a recombination, the flanked DNA between those sites, it has its orientation inverted in 180 degrees. And you can use that to control gene expression. If you think, for instance, your promoter is outside of this flanked region, so if you flip your gene, it's not in the forward sense anymore, so you don't have the proper transcription. And the final model of action is the RMCE, that's recombinase-mediated cassette exchange. That happens when you have two copies of the same site in one molecule and two copies of the complementary site in another molecule. When they are recombined, you have an exchange of the DNA that's being flanked by those sites. And it's a way of integrating a molecule, a sequence in your molecule. And so we've been working in the past years with the inversion as means to control gene expression. We've assembled a lot of plasmids to test the integrase. We were working already in eukaryotic models, and not to repeat the same results forever, we successfully got inversion and gene expression activation when using a reporter gene, and then we moved on to using that on more specific applications in our group. For instance, Daniela talked about the zombie cells, and we used the system to activate the nuclease that's responsible for degrading the genome of the cells. So now we have this kind of mechanism working on different projects in our group, and we thought about ways to expand the use we can do of such molecules in the system. And since our group very focused on application in synthetic biology, and we need, therefore, mechanisms to transform cells, to reprogram the minimal cell, to make it produce something that's of interest. We decided to use an array of such ATT sites implemented on the already existing landing pad on the Syn3B cell. because what we have now is that we can do integration in the system, one event, and that's it. So the idea is to use different integrases sites in a cassette in a way that such one integrase can only recombine their specific ATT site. They will not interact with the other ones. And this way you can have a way of multi-entry landing pad in the system. So the assembly of the proposed enhanced landing pad will be to start with 4 ATTP sites inserted in a plasmid that is able to integrate in the present landing pad using the Krelok system. And upon integration, the idea is to use another integrase mechanism that is the excision, so we can remove the selection marker, since it's another thing that's very precious when you transforming the minimal cell, because you have one that works very well, that's pure mycin selection, and one that works but not that greatly, that's gentamicin. You don't have that many. So if you are able to recycle this marker, it's an interesting way to proceed with the work. Here is just a demonstration of what we expect to achieve. So once it's integrated in the genome of Syn3B, we can add a circular DNA carrying the complementary ATTB site. And when you have integration activity, your sequence will be inserted in the genome, and we can repeat the same process for selection marker recovery, or you can just add a different DNA cargo in the other available entry spots. And for now, the planning we're doing is to use different strategies. The main one I used this as an example before, that is the integration-based system, where you have just one site in each molecule and you have the insertion. Or we never worked with here, but in the literature, it's a very common way of using such integrase. That's the cassette exchange, because one advantage of this model is that you can insert just a part of your molecule. Everything else that's on the outside is going to have a transient expression, and that will be degraded. So that way you can insert, for instance, have your integrase on the outside. It will perform the integration, and then it's removed off the system, and you can use downstream applications. So the plans for the landing pad we're starting to work on are basically that. And another thing that we plan on doing that, but that's not that close future, because it's quite more complex, is to use chimeric proteins in which you're going to have the Cas9 and the catalytic region of one integrase fused together. Because that way you can use guide RNAs to guide your integrase to specific sites on the genome that will work as pseudosites. Here are 2 examples from the literature, when they use different strategies, but to achieve the same end. You can guide your Frankenstein protein close to a pseudosite, so you don't rely on already having the site inserted in your cell. And just to bring an update from some things we're working on the last workshop, that is, since we are always trying to focus on producing things and programming the cell, we need also inducible promoters. So we've tested 3 promoters and 2 different constitutions of one, so like 4 entities. In the minimal cell, there are promoters that are already being tested on all the Mycoplasma strains. 2 models induced by arabinose, 1 and 2 other ones for IPTG. And since John likes negative results exposed, so the IPTG one, they had very different behavior from one another, but none of them worked. One had like it was very leaky, even in the absence of inducer, and the other one had no effect. The one we already have, at least the dose response behavior we expect from an inducer promoter, it's this arabinose induced with the permease and the effector. But an important detail is that when we compare the fluorescence levels of the MTR reporter to the already used tetracycline-induced promoter, you can see that this expression is very, very low. And here we have like around 400 units, while here is like almost 30 times less than that. And one thing that can be discussed is that when they brought the system, the TET system, from Mycoplasma pneumoniae to the minimal cell, the concentrations you use here are much higher. Use here is like 0.4 micrograms, and in the pneumonia, the original works were with nanograms per ml. So we try to increase a little bit the concentration for the arabinose, but the problem is that with 5%, we already don't get growth here. There's inhibition. So to try and get some kind of inducible promoter that can work in the system, we're probably going to try and do some experiments with light inducible promoters, because we don't have this concentration threshold at least, and we hope it works. So that's it for now. Thank you.
So, Marco, I know that you are nearing the end of your doctoral program. Yes. Are you still, I mean, it seems like there's still some wet science to do in what you're proposing. Is this going to be you or the next person coming through to replace you?
I'm going to start doing that, but no strings attached. I can train people very well.
Mm-hmm. Okay.
But yeah, at the first moment,
these could be some terrific new tools that it would be good for our community to have.
Oh, I hope so. In the everyday work and the bench, sometimes we miss this kind of tools. So if we can get to use them here, we're obviously going to share and hope it will help others. The idea is to make the minimal cell a more plastic platform for applications. I mean, all the other studies are applications in a way, but like more direct application like reprogramming and making it produce things.
How good are the light inducible promoters in other systems?
They're not great, but they're controllable. And I think the main thing we're going to try to surpass with that is that concentration threshold, because I'm not an expert on inducible promoters in Mycoplasma or anything, but the experience I've had so far is that for some reason, or maybe different reasons, the minimal cell requires higher concentrations, but they can be very sensitive to the higher concentration. So it can be a problem. If you have one that bypass that, it can be useful, I guess.
Okay. Well, thank you, Marco.